Welcome to Here for a Cure, a series of informative chats with relevant experts in different areas of Parkinson's disease research or therapy. We look forward to sharing with you news of some of the challenges and the latest advances and breakthroughs from around the world. We hope you'll enjoy these discussions. Cure Parkinson's New Zealand is a charitable organisation raising funds for research leading to a cure for Parkinson's disease, the world's fastest growing neurological condition. We define this as the availability of therapies that can stop, slow, or even reverse disease progression, which currently remains inevitable. Existing treatments can only address the symptoms and not the cause of the disease. The good news is there are some exciting new therapies on the horizon, but these require definitive evidence from clinical trials to obtain approval for use in patients. With better tools now emerging for early diagnosis and tracking of progression, we're in a better position than ever before for clinical trials to be successful. We imagine a world where Parkinson's disease progression is no longer inevitable, where people with Parkinson's can live long and healthy lives. We look forward to taking you along on the journey. I'm joined today by Professor Maurice Curtis at the Centre for Brain Research, University of Auckland. Maurice, thank you very much for joining us today. I know you're a very busy man and hard to pin down, so really grateful that you could share some time with us today.
You're welcome. Pleasure to be here. Thanks, Daniel.
Great. So obviously the context we discussed for this first in the series was to talk about some of the recent advances that might have been presented at the AD/PD meeting that you attended at the end of March in Sweden. But on discussion, actually, we thought it might be good just to zoom out a little bit and give some context for our audience about some of the basic biology of Parkinson's disease. Not in too much detail, of course, but just to help people then understand later on when we're talking about some advances around, biomarkers. There was a recent finding, Michael J. Fox funded research that we did a wee blog on, and lots of other stuff in the clinical trial space as well. So maybe that's a good place to start, Professor Curtis, if you'd like to tell us what is alpha-synuclein?
Yeah, so alpha-synuclein is the protein that is most associated with the development of Parkinson's disease. And to give you some context of a cell is made up of thousands of different proteins, and proteins are made up by amino acids. So you eat amino acids, the body breaks them down and then builds them into the proteins that it needs to do its job. In the brain, you've got about, roughly speaking, 100 billion neurons, and all of those neurons are made up of many different types of proteins. And one of those proteins, which is normally involved in allowing neurotransmitter release, so the release of dopamine, and also the release of, also the functioning of the nucleus, or the innermost part of the cell, that's made up of different proteins, but we also find alpha-synuclein there as well. And so alpha-synuclein is a protein that normally helps the cell function as it should every day throughout your life. But for some reason, it gets modified. The protein itself gets modified, and it ends up changing that modification changes the function of that protein. And so it misfunctions or dysfunctions in Parkinson's disease. And we don't know exactly whether or not the dysfunction of that protein is the cell's attempt to make the protein safer for it, or whether it's actually the pathology of the dysfunction of that protein that causes Parkinson's per se. We're still trying to figure that out. But what makes alpha-synuclein so interesting is that normally it has a pretty random shape. But when it gets made useful or functional by the cell, it takes on a very specific shape. But in Parkinson's disease, we know that shape becomes misshapen, and this creates part of the problem that we have in Parkinson's disease. The cell can no longer handle or regulate what that protein is doing. And so it creates dysfunction and eventually cell death, we think, in neurons within the brain.
Right. Thank you. Thank you for that explanation, Maurice. So yeah, we've got an important protein that functions normally in cells that is getting misshapen, and this change in shape, change in conformation is resulting in some problems occurring in the cell, and presumably then that's what's leading to the neurons dying that we see in Parkinson's disease, right?
Yes, that's right. Yeah.
Yeah.
I think one of the interesting things is that alpha-synuclein seems to cause particular dysfunction in only some types of neurons. So many neurons survive quite well with alpha-synuclein in any of its forms there, but some particular types in the brain are really sensitive to this misfolded protein, and hence we end up with some regions that control movement in the base of the brain that end up being worse affected than other regions in the brain with this particular protein. So it's particular susceptibility that leads to Parkinson's disease in those cells.
Right. And you're looking at alpha-synuclein and other tissues as well outside of the brain?
Yes, so there's a big push to try and understand what goes on early on, both in the brain, but also in the rest of the body, when people start to develop those first things that are going wrong in Parkinson's disease. And so we normally refer to Parkinson's disease as being when people have normally the classic symptoms of Parkinson's disease may be a difficulty in being able to initiate their movements, sometimes some executive functioning disappears, their executive functioning goes to a certain degree. Those are the classical signs or symptoms of Parkinson's disease. But years before this, that alpha-synuclein will already have disrupted some processes in the body. So one of them is the sense of smell that goes very early on. But also there can be changes that occur in some of the other places we have neurons, which is throughout the body. And so there are studies that show that you can identify alpha-synuclein accumulating in tissues such as skin and other parts of the other organs of the body that indicate that something's starting to go wrong in the body. Of course, that's pretty exciting because that gives you the opportunity to potentially intervene years before someone loses their substantia nigra neurons that lead them to get the overt symptoms of Parkinson's disease. So we want to be able to identify those things very early on because that's the canary in the mine, as it were, that tells us that something's going wrong. It's a sentinel.
So then, and maybe this brings us back to the meeting in Gothenburg that you recently attended. But in terms of therapeutic strategies, I guess we've got this misshapen protein. We can maybe find ways to help it retain its normal shape, or we can look at strategies for removing it. But what are some of the therapeutic approaches based on alpha-synuclein biology?
Yeah, so there's a couple of really key ones that I think are being explored now, and they're in the exploratory phase. They're not in the full-blown clinical phase at the moment. But the one that was most spoken about at the AD-PD meeting in Gothenburg recently was about immunotherapy for Parkinson's disease. So you've probably seen in the newspapers and in the news about immunotherapy for Alzheimer's disease. And that's where, in the same way that you can immunize against particular viruses or particular proteins in the body, also it's possible to develop therapies that would essentially immunize the body against the amyloid in the case of Alzheimer's disease, or in the case of Parkinson's disease, alpha-synuclein. And so there's a lot of therapies where they are targeting antibodies towards the alpha-synuclein so that the body's own immune system will break down the alpha-synuclein and get rid of it. Now, the challenge with this is that alpha-synuclein being they call it natively unfolded. It means it has an irregular random shape under normal circumstances, makes it difficult to make one antibody that will detect just the alpha-synuclein. And so working more and more towards understanding how alpha-synuclein commonly misfolds is part of the process here. So if you understand what the shape of it is, it's more easy to develop immunotherapies that will target that particular protein. So that's an exciting area, and there's a number of research groups that are looking at that. Of course, that's still targeting the disease quite late on when you get the overt symptoms of Parkinson's disease, and it would be nice to be targeting it years before that. Another interesting therapy, there's a group that has come up with, in fact, this is in clinical trial, have come up with the idea of supplementing the body or the brain with more what we call monomeric alpha-synuclein. So that's the simplest form of alpha-synuclein. And their argument is that as the alpha-synuclein folds into clumps of protein that become toxic or clump together, and we want to get rid of those, as it does that, it uses up the simple form of the alpha-synuclein, and therefore there's no alpha-synuclein around or less alpha-synuclein around to do the normal function that alpha-synuclein should do. And so what their approach has been is to actually supplement more of the simple form of alpha-synuclein so that the body can continue, or the brain can continue to use alpha-synuclein in its normal form. Now, we've yet to see whether that will prove to be an effective therapy, but it's a great example of a different way of thinking about the disease, which I think we'll fairly soon know whether or not that's a good way to be thinking about the particular disease.
Yeah. I'm glad you mentioned, just coming back to the antibodies as well, I'm glad you mentioned the Alzheimer's trials. Those have been a little bit controversial, but it's, there've been some press releases recently showing that they do slow cognitive decline. So a lot of promise there, still a lot of work to do, of course. And I know there have been clinical trials of antibodies with targeting alpha-synuclein in people with Parkinson's disease. But I guess one of the key things, and you alluded to this as well, is just identifying patients to treat at a very early stage, right? Because it's possible that trials are geared to fail in some ways because the patients they're recruiting are quite late stage. They've already been diagnosed with these motor symptoms. They've had quite extensive loss of neurons already. So this is where it's, I guess, another area to look at with alpha-synuclein. There was the recent biomarker breakthrough publication as well. And so, what sort of work is going on in that space around early detection of Parkinson's through perhaps the seed assay that was reported on, or other techniques, but also, I guess, having an objective biomarker for Parkinson's that enables us to track progression as well, so we can determine if therapies are stopping, slowing progression.
Yes, yeah. So obviously, when there's no therapy available or no substantial therapy available, then people don't tend to focus so much on what those early biomarkers are. But biomarkers are great for being able to allow you to see whether or not your potential therapy is being effective, whether it's changing what you are wanting to change. And so there's a whole range of them actually that we know are fairly effective in Parkinson's disease as biomarkers. So loss of the sense of smell is a good indicator in many that neurodegeneration is on its way. Now, about 60% of people who lose their sense of smell and don't regain it go on to develop a neurodegenerative disease. Now, that leaves a lot of people that lose their sense of smell but don't go on to develop a neurodegenerative disease. But often there's other reasons that they've lost their sense of smell. Things like REM behavior sleep disorder, you may have heard about it, commonly known as thrashing about in the bed or living out your dreams while you're asleep. That has a very high predictive value of whether someone will go on to develop Parkinson's disease, and so on. So you put these syndromes together, and actually you can get a pretty good indication of whether or not someone will go on to develop Parkinson's disease. And what's great about those biomarkers is that indicating that populations of cells that are not the main ones that will cause Parkinson's disease are having some dysfunction. Once you get Parkinson's disease, usually 60 to 80% of the substantia nigra neurons, I keep pointing behind my ear here, where those substantia nigra neurons live in the brain, they're dead or dying. And so they're neurons that the therapy cannot bring back. And so if we can find out early on that there's loss of the sense of smell or REM behavior sleep disorder, then potentially therapy could be given before those main substantia nigra neurons disappear. And so that's it would be an absolute win. You detect it early, you treat it early, and people go to the doctor with some other disorder, but not with a movement disorder that we call Parkinson's disease.
Yeah, and that's the ultimate goal, isn't it? To stop it in its tracks before it really develops into what we know as full-blown Parkinson's.
Absolutely, yeah.
So alpha-synuclein obviously as a protein is key to that, but there are other genetic causes of Parkinson's, of course, other pathways going on. So from the meeting you attended, were there kind of promising therapies, other clinical trials mentioned, other therapeutic approaches mentioned of interest?
Yeah, I think for the genetic Parkinson's disease, or where there's a very strong genetic component that's causing the disease. So I'm thinking about people with known GBA mutations or PINK1 mutations, these sorts of very strongly inherited causes of Parkinson's disease. There's individual type therapies around each of those different specific types of Parkinson's disease. But what I would say is that they can be incredibly informative about what changes happen in the brain and how those genes regulate those changes. But across the board, most people with Parkinson's don't have a strongly inherited gene or genes. It's probably a combination of complex genetics and then complex environmental factors that end up causing the Parkinson's disease. So one of the areas that has been very promising is the idea of gene editing. So the idea that you could potentially do something, give a formula that would essentially edit out the problematic gene. And this has become a much more realistic concept with the advent of a technology called CRISPR technology. However, that's not going to be applicable to the majority of people with Parkinson's disease. And so I think we need to look at other reasons that people develop Parkinson's disease. But actually figuring out what is causing it to begin with is probably less important if you know you can stop the progression again by early intervention, which I think will be the future treatment. The way we treat Parkinson's in the future is in the same way that people go to have either a mammogram to detect breast cancer early, or looking for other examinations looking at prostate or bowel cancer. In the future, it would be nice to have a screening program for the very earliest changes in Parkinson's disease. And then you go, oh, yep, you've got these particular things that tell us that this is going to be a problem. And now we start the treatment very early on. Because I think we might find in the future that Parkinson's disease starts a lot earlier than what we have perhaps thought, and therefore an early intervention might be a way of actually curing the population of Parkinson's disease, being able to get in very early.
Right, and it's funny. I mean, you say a bit earlier. I think. I mean, some commentators are talking even decades, right? So, there's some very early changes there that happen a long time before the symptoms are seen. Yeah.
Yes, that's right. Yeah, I'm not sure that we necessarily see the brain changes that many years earlier. I think, in my mind, I kind of think, between 15 and 10 years before, maybe even a bit less than that. So the other changes might be present in the body, things like constipation, loss of the sense of smell, even before the neurons become properly dysfunctional might happen sometime before the neurons become dysfunctional per se. So I think, maybe 10 years might be a good time to be thinking before someone gets Parkinson's, that we could look for these things and then treat them.
I think you used the analogy of some cancers before, and I think that's really apt, actually. There are a number of cancers that in the past would have been pretty much a death sentence that now are very, very treatable through screening processes that can identify these very early, and then treatments to stop those from progressing. And similar to what you were just talking about with alpha-synuclein, some of those are, of course, antibody treatments as well. So,
Yep.
there's analogous treatments there in some cancers. So the hope is that we would have something like that in Parkinson's. And I guess the two approaches you talked about there was the antibody treatment to kind of remove this misfolded protein using the body's own immune system, and that's kind of like the cancer approach. But then also potentially replacing it in a way that it can still perform its normal function. So do you see those as being quite separate, or would it be a balance of those two, or potentially a mix?
No, I think if you look at the way other diseases have been treated over even my lifetime, things have changed a lot. And where once upon a time, for instance, a heart attack would have been a death sentence. These days, they can be treated with stents, with blood pressure lowering medication, with statins, with blood thinning medications, with open heart surgery. There's a whole array of things, and all of those can be applied to one person, or one of those treatments can be selectively chosen for a person's particular needs. And I think neurodegenerative diseases will go that way in the future, where we go, hey, look, there's 6 or 7 or 10 options for treatment here, and we can try them until we find one that really works. So some of it might be about supplementing the brain with some of the monomeric form of alpha-synuclein. That might be one treatment. Another one might be to try and clear the alpha-synuclein away, and so on. So I think in the future, we will have a combination of therapies that will be effective for a range of different neurodegenerative diseases. And that's what you want in a way, because not everyone has exactly the same form of Parkinson's disease. Some people experience more of the executive function loss. Some people experience much more of a movement disorder with Parkinson's disease. And so the concept that you would have one treatment for everyone is probably not the way we'll go. I think there'll be a range of treatments, and you use the one that best fits the symptoms that an individual has. But we need to get over the hurdle of not having a lot of treatment options for Parkinson's disease before we can start down that journey. Because I think we often overlook the fact that actually levodopa, the number one treatment that gets used for Parkinson's disease, is actually fairly effective for many parts of the condition of of Parkinson's disease, but it's not effective for all, and it's not a long-term treatment that leads to a cure. And I think getting in early is going to give us the chance to actually go for a cure, or a prevention of the main symptoms of Parkinson's disease, rather than just trying to treat the movement disorder at the end.
Right. Yeah. And potentially there, you could hold them at that point where the levodopa is effective for a much longer time because the disease isn't progressing.
That's right. Yes.
And look, I'm glad you mentioned that. Levodopa has been around for a long time. And one of the key messages we keep bringing up in our events and so on is the fact that to date, there is no disease-modifying treatment. It's one of the big challenges we have. The only treatments really are about treating symptoms. So, perhaps from the ADPD meeting, any silver lining, any light at the end of the tunnel there in terms of disease-modifying therapies?
Yeah, so there were some incredible talks at the ADPD meeting. I have to say, it was the best meeting I've been to in the PD space. It was just incredible. The 2 drugs that have come on the market, or one hadn't yet actually, for Alzheimer's disease, lecanemab, and another one that's come out since. There was a real hubbub because we were making some progress here, and there were now additional FDA-approved drugs for Alzheimer's disease. And so with that comes a huge drive and a huge enthusiasm, a shot in the arm, as it were, a boost to people's drive to find new treatments. But I think there were a number of them looking at a number of people talking about the structure of alpha-synuclein and how to target early on, stopping the misfolding happening. So a bunch of drugs called disaggregases while also looking at which slow down or stop the misfolding or aggregation of those proteins. That was certainly a hot area, and particularly for Parkinson's disease. And then also a whole bunch of studies were presented that looked at what the conditions are that are required to cause the protein to misfold. And so again, this is looking at ways that humans in the future, people in the future, will be able to potentially have preventative approaches to getting Parkinson's. Now, we're not there yet, but this is the very thing of how do you prevent those first initial events happening that lead to these major events like Parkinson's or big clumps of alpha-synuclein down the track from happening. And so those are the types of things that I think we've always seen a little bit of that, but this was really on show at this particular conference, and there was a real hubbub and drive around it. Lots of questions, lots of young scientists presenting their work in some of these areas. It was really exciting.
Oh, that's fantastic, and it seems to me a really exciting time to be involved in this field. I think we're certainly getting a lot closer to our dream of a cure or cures for Parkinson's disease. So.
Absolutely, yeah.
Thanks.
Yep.
Thanks so much for that update. And just finally, before we go, Professor Curtis, any updates from your lab, your research at the moment? How are things going over there?
Yeah, no, we've had a pretty busy year so far. It's been a very good year, and we've got two major pieces of work that we've actually got one submitted, and one of them is the work that the Neuro Research Charitable Trust, Cure Parkinson's New Zealand have funded, and this is the work looking at the olfactory bulb structure in humans and the olfactory mucosa. So the place where we believe and we have evidence for alpha-synuclein showing up first in the brain. We've submitted that work now. It's under consideration at a big journal. So we're really excited about that. And it's been a pleasure over the last couple of years to show you the advance in that work. We certainly are over the moon with how that's come together. And then also we've had the opportunity to actually work on a bunch of about 110 samples from people that had donated tissue from the roof of their nose and also from the olfactory bulb, to allow us to identify these deceased individuals that will allow us to identify alpha-synuclein and at what early stage that might show up in a population that doesn't have Parkinson's disease. And we've been working a lot on that. In addition, we've got a piece of work that we will have going out very soon, which is looking at the regional distribution of alpha-synuclein in the brain and how the misfolding pattern looks in different regions of the brain. So there are two exciting areas that I hope to be able to report back to you shortly on how those are going as they make their way out to the journals for the world to see.
Oh, that's absolutely wonderful, and we look forward to seeing those in press, of course, and really do appreciate your updates. And it's just wonderful that NRCT, Cure Parkinson's New Zealand, can help support this work in any way we can. So,
I really appreciate the support. All of my lab members here do, and we just need to get closer and closer to that early detection and cure.
Yeah, all of those together, right? Yeah. Well, look, thank you so much, Professor Curtis, for your time today. It's been absolutely wonderful to get that update from you. We really don't want to distract you from your work, but really appreciate that and your time. And we're also very much looking forward to having you at our fundraiser dinner in August this year. So thank you so much.
Can't wait. Thanks, Daniel.
Cure Parkinson's New Zealand is a New Zealand-based charitable trust raising funds for research leading to a cure or cures for Parkinson's disease. We are the only New Zealand organisation solely focused on Parkinson's research, and we collaborate strategically with like-minded organisations globally, as well as other funders of research here in New Zealand. We rely on donations and generous gifts to make this happen. If you would like to support our mission and play a part in bringing the first ever disease-modifying drugs for Parkinson's to patients, please get in touch for a conversation about how you, too, can help make our vision a reality.